Hello everyone. Hope you guys are fine. Did you all downloaded those files in your system?
Yes.
Okay, everyone?
Yes, ma'am. Yes, ma'am.
Oh, okay. So have you made one folder with the name raw data? In the raw data, you have to keep those files, okay? Make sure.
Yes, ma'am.
Yeah. So we will just wait for another 5 minutes. Meanwhile, people can join. Then we will start. Can you see my screen, guys?
Yes, ma'am.
Okay. So our very first step is to see how to download the data, right? So you just have to go to the NCBI SRA. So we are talking on the RNA sake, right? So I will just write here RNA sake and search it. So now after searching for this RNA sake, can you see there are a lot of hits for this RNA sake below? And now if you want to download anything like any for specific organism, you can select that. It's up to you from which organism you need the data. You can select that from here. And on the left hand side, yeah, you can see there is a option for the assess. So first is the controlled. So controlled assess means whoever has submitted the data, they don't want to share the freely. Okay. So we have to ask the permission, like, okay, ask for the permission to download the data. Whereas in the public means it is freely available. Okay. Now, again, the source, it's up to you whether the DNA, RNA, you can select that. Type, exome, genome, and library can be of paired also, single end data also. It's up to you. If you want to work on the single end data, you can just select that. This many entry will give you for the single end. Then again, on which platform you want to work, whether on the Illumina and Torrent, these are what the sequencing platform. Okay, they are mentioned here. And the strategy. So whenever like we are choosing the RNA-seq strategy, okay, the source you will get what the transcriptomic, okay, and the data it is like they have downloaded in this GSS3 format, and the file types you will get in this format. So now I will tell you from where the data has been taken. Can you see this experimental? This is the ID and the title of the project. RNA-seq analysis of human lung cancer cell line. Okay. And they have worked on the Lumina machine. How many times they have run and how many spots they have used, they have mentioned here. And what is the size of this data? You can see here. Okay. And who has submitted this data? That university name they have given, and this is the study title, you can say. And if you want to study more regarding this article, you can see the abstract from here. At your home you can study this the abstract. In the library you can see they have used the Lumina platform. The strategy is the RNA-seq. So I told you, whenever the strategy is the RNA-seq, the source will always be the transcriptomic. And now this data is what the single end data. Now, if you see here, can you see the organism? The organism is human. So this data has been taken from the human. And if you select all runs from here, it will just navigate to this SRA run selector. Can you see the IDs here? And this is the control one. And this is the treated. Okay. Why this is control and treated, that you can see later on in your the abstract part. You can study the abstract, you will come to know why it is control and this is the treated one. Okay. So like, did you notice in the folder you were having two control files and two treated files, right? So which control I have taken? So you can see this is the 458 and 459. This is the control one I have taken and this is the control two. It's up to you which data you want to select. You can select that according to your choice. And the treated one I have taken this one. This is my treated one and this is my treated two. And how to download this data? So for example, you can just select this entry and you can just download from this also. You can see FastQ. Okay. But I always prefer the ENA database. You just have to put the IDs and just view, and you will again get all the same information. You can see the single end data, RNA-seq and transcriptomic. Below you can see the link they have provided. If you want to download this data in your terminal, you just have to copy this link from here. And I will tell you the commands, what it is. So wget, then space -c, then put the link. So I will just write here wget -c and I will paste that link. Okay. So from here, wherever the data like you have created, There itself you have to open the terminal. So you just have to type a wget -c and then paste the link. It will start downloading that control 1 file. But you can see here the size of the file is very huge. If you see in the NCBI SRA. Can you see this GB? It is there, right? So what I did in my data, I have cut down the 50 lakh reads from the control and the treated one. Okay, so how I have cut down my data? Just putting the head command, then -50 lakh, then put the ID of your data. Okay, then this is my output file which I have shared with you guys. Why I have cut down the data? Because some of the system won't accept this huge file, because this is the huge file, it won't run easily. Otherwise it will take lot of time to complete this RNA-seq application in the system. Okay, you need the server actually to run the original data. Okay, but for you guys I've just cut down only this 50 lakh reads and I've shared this files with you. Is it clear what
Yes, ma'am.
this control 1? Okay, so now this is how we are downloading the data. Okay, now after downloading the data we need to check what the quality. Correct, so everyone is having the fast QC tool in your system.
Yes,
Uh,
ma'am.
Yes,
yes,
ma'am.
ma'am.
Okay, so what you guys have to do, so just open that raw data folder where your files are, this files, control 1 file and created files. So from here you have to open what WSL. Okay, opened,
Yes, ma'am.
and you just have to write ls. Ls will list you all those 4 files. Can you see that on your screen? 4 files, control and treated
Yes, ma'am.
files. Okay,
Yes, ma'am.
now you have to open the manual for the fast QC to check whether that tool is installed properly in your system or not. Okay, you need to open the manual. Just type fast QC -help. If this manual is opened in your system, that means fast QC is installed properly in your system. Is it opened?
Yes, ma'am.
Okay, so now in this manual, you can see how to run this tool. You can see you just have to put fast QC and then you have to mention your file names. Okay, so what you guys can do in the terminal, just write here touch RNA sec pipeline .sh. So it will open in the notepad. Just try this command, touch because we want to create this script. Can you? And every command you have to write in this script, okay, in this pipeline. Is it opened in your system?
Yes, ma'am. Done.
Yes, ma'am. It's open.
Yes.
Done. Okay, so very first step you have to write here this, that is this hashtag 2. I will just share this command, okay, in the chat box, but just write this quality control. And the command I will tell you. So first command is fast qc and give the file name. What is my file name? Control 1 underscore dot fastq, right? Another file is control 2 to fastq, then treated 1 underscore dot fastq, then another treated.
Ma'am, we can use star dot
I will
fast.
tell you. I will tell you. I will tell you. So for the quality control, if there is a two file only, that time you can use, you just have to mention fast qc and give your file names. But you can see if what if I'm having multiple files, right, of the fast queue. So can you see there is a common extension here, dot fast queue, in the control 1, control 2, treated 1, right? It is common in all the files, right? So in the module 1 in the Linux command, you might have heard about the star, right? So star is indicating what? The all, right? So you just have to write on the terminal fast qc, give the space star dot fastq. Why dot fastq? Because it is common in all the files. Then just copy that and then run on the terminal. Can you? One of you guys can share your screen so that I will be sure like you are able to do that. You all have to run this fast qc in your system. Okay, it will take a time. Just share your screen, one of you.
I'll do it, ma'am.
Yeah, sure. So in this steps, we are going to check what the quality of our raw data. Raw data is what? The control and the treated file. Okay. Yeah, correct. Can you see guys, everyone on a system? So it is running. Can you see that? It is has been done for the treated one. It is doing for the treated two. Likewise, it will do for the control one and the control two. When it is done, okay, then let me know.
Yeah, ma'am.
And make sure all of the command you have to write it in that pipeline only, okay? Because in future if you want the reference you can just see that. Yash, Sonali, is it working in your system?
Yes, ma'am. Done.
Yes, ma'am.
Okay. There is Sanya also, Namarta, Juhi. What about you guys?
Okay. It's like I have two doubts. So
Yeah, yeah.
you told us that you usually prefer ENA
ENA, right?
database. So why is it so? I mean, why
I will
not?
tell you. Okay. So you can run this on terminal. Meanwhile, I will tell you why ENA. Okay. Let me share my screen. Can you see my screen?
Yes.
Okay. So, ma'am, it is fine for the single end data if you are downloading from the SRA also. The problem is when you are downloading the paired end data, you won't able to find out whether it is forward strand or the reverse strand. Okay.
Okay.
So I will tell you how it look like. So for example, I want to work on paired end data. I'm just selecting this paired end. And make sure never download or prefer this latest one, this first 10 entries. Why? Because still the research is going on. Prefer always the previous or the older data. So what if I just write here 6000? So from where I was downloading, I was just going to all runs. And if I go down and I want to select one of the entry, just go to this fast queue. Can you see here? You just have to directly download this. But here you can't say this is either the forward stand or the reverse stand. Right?
Yeah.
But if I just copy this ID and go to this ENA browser and just paste that, same information you will get that. This is not the ID. Let me see whether it is the paired and data or not.
It's a paired one.
Yeah. Yeah. Can you see when I put this data in the ENA, can you see underscore one here and underscore two?
Mm.
So
Yes.
this will give you what the forward stand and this will what the reverse stand here, ma'am.
Okay.
But in the SRA, you won't get that. So there is a long procedure in the SRA. You have to run lot of tools to separate those forward and the reverse stand. But still, like there will be the error also. You might delete some of the data also, right?
Okay.
So never prefer the paired end data with the SRA. Okay, so you just have to select the data from this SRA. Okay, and then just select or maybe copy that ID and go to the ENA. They will give you separate. Again, if you want to download this data, you just copy that address. And how to just write that? Wget -c and just paste that link. It will download that file in this terminal. Okay, clear?
Okay. Yes. Thank you.
So fast QC, it is done in your system, guys.
Yes, it's done.
All of you?
Yes, ma'am.
Yes, ma'am.
Okay. So can you see in your system you are getting two files for the control one, that is the HTML file and the zip file. Can you see that? Open the folder in your system.
Yeah, ma'am, it's there.
Yeah. And this zip file is containing what? The summary of the graphical representation. Whereas this HTML file is the graphical representation. If I open this HTML file, can you see the QC report of your control one file?
Yeah, ma'am.
So in this QC report, can you see there are a lot of options on the left hand side? So on the left hand side, the important part is that you have to check this per, you just mention, like you can just note somewhere, per base sequence quality, over represented sequence, and the adapter content below. There is this one. So these are three important parameters. Okay. Whenever you are just opening the QC, you just have to focus on this three parameter. So what is this green tick, red, and this exclamatory? So whenever you are just getting the green tick here, that parameter is a good quality. Whereas this exclamatory is what? The data is having the moderate quality. So it is having the average quality, you can say. But if you're getting a red cross, that means what? Data is having the bad quality. Okay. So you can see here in the file name, it is showing you what? For which the raw data you are checking the quality. That file name it is giving you. That you can see control1.fastq. Next is the file type. File type is what? The actually base calling method they have used for each and every base. Next, encoding. So the Illumina platform or the Sanger platform they have used. The total sequence means the number of reads in the sample. Sequence flagged is 0. That means out of total read, no read is having the poor quality. That is why they are giving you the 0 number here. And next is the sequence length. And next is the GC content. Okay, in your data. Here you can see in the per base sequence quality. So whenever, can you see this line? Whenever it is falling in the green color, it is indicating what? It is having the good quality. But whenever it is in, like the bases are falling in this orange one, that is having the average quality. And when it is falling in the red, that is having the bad quality. Okay. And this y-axis is indicating what? The Fred quality score. Clear? So we have discussed about the good quality. We need to see the bad quality also. I will show you. You can see the bad Illumina data here. Can you see the red mark here? So can you see this yellow boxes? Yellow boxes is representing the bases here. Okay. And these boxes are actually the whisker plot. Actually, it is divided into three regions: upper, lower, and the midden quartile. So you don't need to worry. So quality control, all it is having just the algorithm. It is just making by itself only. Okay. And now you can see these bases are falling in the red color and the orange, right? That is why it is giving you the red cross. That is what the bad quality. So never prefer this type of quality in your data. So if you are getting the red cross here in the per base, overrepresented or adapter content, that means you need to change your data. Okay, you cannot proceed further. So here in the overrepresented sequence, I want to tell you. So if it is giving you no hit, so that is fine. Okay, that you can go ahead with these sequences. But if I scroll down, can you see this Illumina paired? So whenever the primer, can you see this primer, Illumina platform, it is there mentioning. So that means that particular sequence is what? The adapter. So that sequence you need to remove from your data. We are not interested with that. And now if you want to study in detail what are these, okay, that also you can study at your home. Just go to this copy of this fast QC analysis module. One by one you can open and it will tell you what it is including. Okay, can you? Any doubt till here in the QC part?
No, ma'am.
Now, if you guys open control 2 file, treated one, you can see each and every file, it is having the good quality. Can you see per base sequence quality is also green, overrepresented? You can see there is no adapter sequence here, right? And adapter content here, it is just equal to 0. So that means it is not having any adapter sequence here. Also in the treated one, you can see overrepresented sequence is also not there. So you do not need to go with the trim, right? Can you see that? But if you find this type of data, so that time you need to trim that sequence from your file. So how to do that? I will just share the commands with you and just I will discuss how to do that. So we are just cleaning our data with the help of this cut adapt tool. So it's not always the cut adapt. It's up to you which tool you want to use. Okay. Which tool is more comfortable, you can use that. It's up to you. You just have to simply install the tools in your system and just open the manual. It will just give you all the commands in that. But for now, I will tell you what is this cut adapt here. So this is basically for the single length. How to run that? You just have to mention cut adapt, give the space and -b. So here in the -b, I have mentioned sequence, right? So for example, in my data, this is my adapter. You just need to copy this and here you just have to paste that particular sequence. Clear? Clear? Then
Yes.
-q. So -q is what? The Fred quality score. So I always want Fred quality, it should be 30 or more than 30. Correct? So what you have to write 30, 30 here. Then -m is for what? The minimum length. So there are some reads which are having the length like less than 20 also. So I'm not interested with those reads. So that I will automatically delete with the help of this M. And this is -o is output file. So it's up to you which file name you want to give, you can give. And this is my input file. If that particular sequence is present in the control one, that input file you have to mention here. Clear? How to run this tool? Now, whereas in the paired end, you have seen in the ENA, it was giving you the -1 and -2, right? In the paired end, you are having what? The forward data and the reverse data, right? So in the paired end data, you just have to write cut adapt. And in this -b, you have to give a sequence from the forward file. And in the capital B, here you have to put what? The reverse. Okay, reverse file sequence. So whenever there is an adapter in the reverse file, that time you have to paste here. Again, this is the Fred quality score 30/30 -m minimum length. So it's again up to you which according to which length you want to have. You can just mention that. This is my -o. -o is again the output. Clear? For the forward. But if you want the output for the reverse file, that time you have to label with the -p. So you can see I've given the output file name, that is this. And then further you have to put the input file name for the forward and the reverse. Clear?
Yes, ma'am.
Now see, after doing this trimming part, you need to do the quality check again. We need to see whether that particular sequence is removed or not. Correct? So what we are doing again, we are checking what? The quality. So what will be the command? Fast QC. Can you tell me what are the input files now? One of you can tell me. For which quality I'm going to check now?
Ma'am, for the trim control one, for the trim control one and trim control two.
Nice. Correct. Now this, it is become what? The output file name, right? So whatever the data would be there, this is will be the my output. So this thing, it become what? The input file, right? So after running this, you will find this particular sequence will get deleted from our data. Clear till here? Now, but like in our present data, our data is good. So you don't need to trim our data. Okay. Now, directly we are going to map our reads in the reference genome. Correct? Next step is what? The mapping. But before mapping, you need to download what? The chromosome, right? Or the reference genome. So I've already studied the abstract. So that particular gene, okay. So this data has been taken from human, correct? So if I will download the entire genome of this human, your system gets stuck in between. So that is why this gene, okay, it is present on chromosome 16. Okay, so that is why for now we are going to download what? The chromosome 16 only. So how to download that, I will tell you. Just go to the UCSC. Along with me, you can also go. And there is an option download. Just select that genome data from here. And there is an option human. Because our data is from the human, you just need to select that. Again, the assembly should be what? The updated assembly. So I won't go with this because it is not giving the complete genomic information. It is just to telomere to telomere. I will go with this entry. Okay. Now I don't want to download the entire genome, right? Otherwise it will take a lot of time to run the pipeline in your system because that much configuration, it is not there in your system. Okay, that is why we are dealing with the demo data. Okay, so just select this data by chromosome. Here you will get that chromosome 16 for the human. Just go down and you will get that. Can you see that?
Yes, ma'am.
Right, you just need to copy that link. Okay, then you just have to write wget -C and paste that link and try to open that on terminal. Can you just download the chromosome 16 from the terminal? Did you got it? Why we are downloading only chromosome 16, not the entire genome of human? Everyone, it is clear?
Yes, ma'am.
Okay.
Yes, ma'am.
Okay. Just try to download this chromosome 16. Should I share, like copy this?
Done, ma'am.
Done? Can you just share your screen? Everything should be downloaded in the raw data only, okay? In the RNA-seq folder. Okay, nice. Rated one. Control you are getting. Yeah, great. When it is done, let me know.
Done.
All of you, it is done.
Yes, ma'am.
If anyone is stuck in between, just let me know. Don't hesitate, okay? You just perform the commands. Like at last, I will just share with you guys, okay? You don't need to worry with this script. I will just share all the commands with you. Done?
Done, ma'am.
Okay. Sanya, Arya, what about you, Juhi? Is it done? So,
Done, ma'am. Done.
okay, fine, fine. So now in your system, like in your folder, you can see your genome file. It is in the zip format. Can you see that extension in your file .gz?
Yeah, ma'am.
Right. So we need to unzip that. Okay. So for that, a command is g unzip and put your input file. Okay. The file which you want to uncompress it. Can you just try it? G unzip.
Done, ma'am.
Done. Nice. Okay, so now we have downloaded the genome also. Our next step is what? A mapping of the reads, right? So in this mapping, you are having the two steps. first step is to create your index genome. So, any idea why we are indexing our genome? Why we are not directly mapping on the genome?
No idea.
No? Okay. So you can see this genome file, it is given in the FASTA format, right? Means it is in the human readable format. So for the mapping, we are going to use the HISAT tool, right? So computer won't understand that format, right? Because that is in the human readable format. So that is why we are going to convert this. Okay, we are going to index that. So this is the first reason. And second reason is that whenever we are doing the indexing, so it is creating what is short binary files. So it is just reducing the time of the mapping. Okay. So it will, what happen, like whenever you are having the short binary files, it won't take that much time. But if you take the entire genome, it will take lot of time. Just to reduce the time, it is we are just indexing our genome. Clear? Why we are indexing? So first thing is we need to install the HISAT2. So everyone is having HISAT2 tool?
No.
No?
No.
Okay. So just open the terminal and just write here sudo apt -get install HISAT2. Just install that tool. Done. And after that, you can just write here HISAT2 so that you will be able to know like whether it is installed in your system or not. Okay, manual will be opened.
It's installed, ma'am.
It is installed. Okay, everyone, like it is installed in your system.
Yes. Yes, ma'am.
Okay, fine. So after installing this tool, we are going to index our genome. So very first step is you just have to call your tool, that is the HISAT2 tool. Okay. Now this chromosome 16.fa is our input file. Okay, that input which you have downloaded from the UCSC. Clear? And this chromosome 16 is my now output. And this chromosome 16 will be what? My index genome now. Clear? So here we are not mapping. We are just indexing our genome. And after indexing, you will get these files in your system. Can you see this? 8 binary files. So this 8 binary files computer can read easily. Then further, we are going to map our read on this short binary files. Clear? What we are doing in the HISAT2?
Yeah, ma'am.
Okay. So meanwhile, you can just perform this HISAT2. On the same terminal you need to perform, okay? And again, it's up to you. You can change the output name also. You can write here genome also. Just make sure you should remember those names. So what are the output and the input? You should know that. But for now, I have just kept that chromosome 16. And when it is done, just show me in your system. And further mapping part and the counting of those reads, we will discuss tomorrow.
It's done.
Can you show me those binary
Ma'am,
files?
ma'am, I'm working on different Linux system.
Okay, you are working on Linux system, na? So it's no worry. You can
No,
just
no,
show
ma'am.
me.
I'm actually attending this meeting in Windows and working on Linux system.
Okay,
Different. That
okay,
is on a different
fine,
system.
fine, totally fine.
Yeah, done, ma'am.
Any doubt till the creation of this index genome? Any doubt?
No, ma'am.
Is it clear like it is running very smoothly, this pipeline? Because every step is linked with the another. Okay, then only you will come to know what we are doing in this pipeline. Okay, so any one of you, like if any doubt is there, you can interrupt me in between. Okay, and I will tell you if it is
Ma'am,
fine. Hmm.
are you going to show? It's a bulk RNA-seq, right? So, are you
Yeah.
also going to show single cell?
Hello.
Ma'am, are you also going to show single cell RNA-seq?
No, ma'am.
Okay.
Yeah. But you will have a idea like how to perform this. Once you are able to run this one application, now you can perform any other application also. Okay, because it's just to know how to run the commands. So it's simple. Like first thing is important to install the tool. If you are able to install the tools, then you can open the manual. In the manual there are commands. Okay, and even like if you are very new to this also, you can browse it like how to run those pipeline. But one, if it RNA-seq it is completed, that means you can like perform another pipeline also with yourself.
Okay.
It's not that much difficult. So can you tell me why we need the GC content?
Ma'am, for every species, it is like fixed. If there is some issue
Mm,
with that, we can know that there is some issue in the sequencing.
okay. So that is not the reason actually. In this GC content, what happened like it is giving you the stability in our sample. More the GC content, more the stability in our data. Because you know G is it is interbonded with the three hydrogen bonds, so it is giving you the stability more in the DNA, right? So that is why here it is calculating what the GC content. Got it? So here you can see this bell form shaped. It is already there. Okay, it should be like this bell formed, but still there is a difference. Can you see there is a gap? So this red is for our data. Okay. So if it is properly like the bell shaped, so that will give you the green color, but still it is giving you the average quality for this GC content here. So now can you guys tell me what is this N content here?
Ma'am, while sequencing, if a base is not known, so the computer uses a code like N.
Correct. So N is what basically the gap in our data. So let's consider during evolution, if small stretch of sequence get deleted, so that time it will create a gap. So whenever we are going to do the sequencing, that time sequencer won't be able to read that actual base. So that is why it is giving what the N content. So here you can see there is no gap in our data. That is why it is nearly equal to 0 here. Got it? Clear?
Yeah, ma'am.
Okay. So yeah, we can meet tomorrow then, and we'll discuss the mapping and the counting of reads tomorrow. Okay?
Thank you, ma'am.
Okay. Bye bye. Take care, guys.